Good morning. I want to thank Dr. Lama Moussaoui for including me in this very inspiring program. This is really a very moving experience for me to be back here in Beirut, having grown out of a university from Beirut, and coming back and helping to inspire women. I think this is a great mission, and I really congratulate you for putting this together and for the other organizers. So what I'm going to do today is talk to you about the field of study that I work in, which is quite technical and quite prone to producing lots of data, and actually prone to even kind of merging of data from different technical fields. So this is, in a way, a good seed for people who are interested in science as a kind of way to start, and there are many potentials to expand from small data sets into big data. So hopefully I'll be able to give you a little bit of a flavor for it. In 15 minutes is not a lot of time, but so as you heard, I'm currently a professor of psychiatry and vice chair for research at Stony Brook since 6 months ago, having come from Columbia University for where I maintain an appointment, and I was for 20 years. Okay, so I'll concentrate on positron emission tomography, and it's really kind of an example to lead you into the type of big data science that you may have to deal with within one area of healthcare, which has to do with the brain. So PET, positron emission tomography, is a way of looking at molecules inside the brain, and to do it, you need few pieces of equipment, lots of money first to buy these equipments. A cyclotron that produces radioisotopes. So these are unstable atoms that emit radioactivity to go to a more stable status. By emitting these positron, which are positively charged electrons, they kind of hit a negatively charged electron. They annihilate, and annihilating, they produce energy. We call these photons. Those two photons hit detectors. And basically, PET is about detecting the simultaneous hitting of two opposed detectors, and figuring out that, well, an event has happened along this line. And so basically a scanner is a ring of detectors that detects all these events that are coming from different lines of responses and putting them together. This is what a PET scanner looks like. And the head of the patient is inside that hole. Another important, so PET has a lot of technicalities to it. It's really the ultimate multidisciplinary science. So you need to be able to have physicists to kind of process the radioactive signal. The detectors are hooked up to computers that are going to process this radioactive event. But you also need chemistry, radiochemistry, because the radiochemist is going to take the radioactive isotope that is produced from the cyclotron and tag it onto a molecule of interest. And the molecule of interest is determined by what is it you're interested in looking at the brain for a particular disease. So for example, you want to look at the dopamine system or serotonin system. These are chemicals in the brains with like various receptors by which they produce their effects inside neurons. You decide on which receptors you're interested in. You take a ligand that has specificity and selectivity for this receptor, and your radiochemist put onto it a radioisotope. So then you have a radioactive ligand, which is a radiotracer. So when this radiotracer is injected into a subject, this is how basically we do PET imaging, the radiotracer will concentrate on the sites in the brain for which it has this high selectivity and affinity. And so it's going to give the radioactivity from those sites. And by tracing back where the signal is coming from, you form a map of where the radioactivity is distributed in the brain. So this is where the targets are that you're looking at. Okay? As you produce an image like this. The pointer here, let's see which. I don't know where the point. Oh, do you see? It's very. I don't know if we can dim the lights here a little bit, but so the image on this side is a PET image. Now this is just the beginning of it, and an image is meaningless, especially if you're really trying to understand something about the brain. Now, in cancer, sometimes they can get away with images. That's what the radiologists do. They'll look at an image, they'll see a hot spot, they'll say, oh, this looks like there is a tumor. But for understanding the neuroscience type of questions that we have about the brain, images are worthless. We need to analyze them and quantify them to derive estimates of what is it we're measuring. And so there's a lot of, so I've pointed out to you, there's physics, chemistry, and now there's a lot of mathematics. So really, in my team, I have a full-time mathematician. And that's what he does, is process the data and analyze or oversee a team of people who analyze the data. One particular initial point is how to measure the signal that we have in the brain's radioactive signal is first to draw regions that we want to concentrate on that are encapsulating the radioactive signal and compared to regions that have less radioactive signal. So in order to do that, we have to get an MRI on the same subject. Now, you could also use a kind of an anatomical template that is derived from an atlas or from different people. So we draw the regions on the MRI and then transpose that onto the PET. So a lot of steps involved in this operation. You have to so you have a kind of acquisition over time for the PET images. All these have to be put kind of together so that you correct for movement between them. And then you have to put them together with that MRI so that everything is in the same plane, so that the region that is drawn on the MRI can be transposed onto the PET. And then these different regions, you have the activity over time in each. We call these time activity curves. So you can imagine already like the big data sets that you're getting from all these different regions. And you could even have it from different voxels in the brain. So the smallest units of volume. So there could be thousands of voxels. We kind of like to look at regions of interest. The second big analytical step is to quantify the input function, which is what goes into the brain, the radioactivity that's going into the brain. We need to estimate out of this what is the actual free parent radio tracer and what is metabolites, and all this goes into the calculation. And so for that is another kind of chemistry, data analysis, big operation to figure out how much at every time point of the radioactivity is composed of radiometabolites versus the parent radio tracer, because it's really the parent tracer that is bound to those sites in the brain. Once you have this input function, where is the pointer here? I don't see a pointer. Should have. Okay, so this is the input function. This is really what is getting into the brain, what the brain is exposed to. And the response of the brain is all these time activity curves. So we need to put these two together to estimate the number of sites in the brain that we are interested in. And for that, we do kinetic modeling. Okay? So what is kinetic modeling? This is really the ultimate mathematics. So we basically have a signal that is like this. That blue line with dots is the total radioactive signal we're getting in the brain, but that is composed of different elements. There is specific binding to the target and non-specific binding, just kind of radioactivity sticking around to things that aren't relevant. And we need to tease these two apart using mathematics. So we assume different compartments. These are spaces where the radioactivity is well stirred at all times, so they're homogeneous. There are 3 compartments, for example, for analysis of receptor data. The plasma is one compartment, and from there, the radio tracer is going into the brain where there are 2 compartments, specific and non-specific. What are the differences between specific and non-specific? Specific binding is really the binding of the tracer to the target of interest, and it's saturable. you have kind of a period of association and then dissociation. The ligand and the receptor are kind of binding together, and then you have a decrease in this binding. While the non-specific binding is just non-specific, so as much as you have radio tracer input into the brain and presence of radio tracer, that is going to increase with time. It's linear. So these are some of what I've mentioned about the properties of these two. And basically what the kinetic modeling will do is derive a binding potential that is an estimate of the number of targets in that brain region. And so I'm going to skip the mathematics here to tell you that really within an experiment you need to reach that point of equilibrium, and you also need to be at a tracer dose. So there are specific technical issues to derive the outcome measure, which is Bmax over Kd. Bmax is the maximal number of receptor sites in that brain region. So you just see that you have all this operation to basically get to these numbers, because we don't want images. We want to know how many of those targets are in a brain region. This is an example of if you're looking at the 5-HT transporter. The 5-HT transporter is the target in the brain to which drugs like Prozac bind. You may have heard of Prozac or Paxil. You know, these are antidepressants. And so these are two different tracers to look at the same transporter. So you see differences in the signal, you know, the radioactivity coming from different brain regions. The actual this is the actual area where you have concentration of these targets. You see this is non-specific binding, and you see the differences between tracers. So PET imaging has a lot to do with optimizing the signal to background ratio to select the best tracer and then be able to analyze it and give optimal results and look at the reliability, reproducibility, validity of the findings. That's the first step. Once you validate these, then you can use them to answer clinical questions. Is there a problem with this target in a specific mental disease? Or does a drug bind to this target? So this is really kind of the entry point to look at interesting clinical questions. Let me skip that and just tell you, there are lots of tracers out there, so depending on what is it you're interested in, you can select the one that will answer your question. Tracers for the dopamine system, the serotonin system with all the various receptors and transporters, and less used other receptor systems such as the opioid, cholinergic system, glycine, GABA. And the one important area that is really booming now, and that may be a topic that some of you will deal with, is amyloid imaging. Imaging for dementia. This is one of the recent FDA approvals of use of PET imaging to see who is at risk to develop Alzheimer or other types of dementia by doing a PET scan to look at the accumulation of beta amyloid in their brain, or tau. Okay, so these are really important because as the field generates lots of these images, we need to kind of merge these together and define criteria so that the criteria then can be applicable in the clinic easily. So, we need to understand the specificity and the selectivity, but also how much of it is giving you, what is the threshold of false positives and false negatives, so big kind of biomarker development. So just to not take too much time, this is an example of the type of targets within a dopaminergic synapse that we've been concentrating on to understand one of the basic pathologies in the brains of patients with schizophrenia. This is also relevant for brains of patients with addiction. So you can look at, for example, an enzyme along the dopamine synthesis pathway. You could look at the vesicular monoamine transporter, which is what takes the dopamine produced and puts it in vesicles and stores it in the presynaptic nerve terminal. You could look at some of the receptors, either postsynaptic or presynaptic, the transporter, the dopamine transporter. So there are tools to look at different targets. And like if you suspect, for example, in Parkinson, they lose 80% of their dopamine neurons. So they have a degeneration of dopaminergic neurons. So you could look at their dopamine transporter, which is a marker of how many dopamine projections are there in the striatum, for example, and you'll have a major loss. This is one way of using this type of technology to answer a question that is important for the clinic. And can you reduce this loss when you give a certain treatment? I'm going to skip my own research on the dopamine system, all the findings we've had. This is an important application. When drug companies are developing a new drug, they want to know whether it hits the target. It gets to the brain and it binds to the receptor that's supposed to bind to. PET is absolutely necessary as a screen. You can start by using a non-human primate. You have a tracer for that target, and you give that cold drug that the pharmaceutical company is developing. And if your radioactive signal decreases, it means it's occupying the target. As you see here, you have the initial activity, and then as you increase the dose, you have less and less activity until you really block it completely. So you have full occupancy of that target. And that helps drug companies to determine what is the dose needed that produces sufficient occupancy and is clinically effective, okay? So examples of like really large data sets that PET will either produce or be directly related to, and that is when you analyze looking at every single voxel in the brain, that produces massive data sets, and especially if you're looking at longitudinal time points. And if you're looking at lots of, a big sample size where you have to homogenize all this data to analyze it together and put all the voxels on top of each other across time points and across patients and across groups. Merging with another kind of imaging data, fMRI data, also represents and produce huge data sets. And, computer scientists are badly, badly needed in this area because it's the way to the future. We understand a lot more about cancer than we understand about the brain. The brain is a black box still to this point, and especially everything that has to do with behavioral problems. So I think that for the future, I really hope a lot of you will be inspired to go into neuroscience, neuroimaging, and there's a huge opportunity for biomedical, computer, mathematical type of research there. Developing biomarkers is a great example also of big data sets. So for example, if you go to the ADNI website, the ADNI website is a kind of consortia of different sites of imaging looking at Alzheimer's disease and trying to develop biomarkers that are used in the clinic or imaging biomarkers. And they have great criteria as to how to acquire the imaging data so that it can be pooled across different scanners, different sites, and how it's going to be analyzed so that it's all kind of homogeneous, cohesive, and can produce reliable set of criteria that we call imaging biomarkers to define, who has amyloid, who has tau, and who is kind of basically predicted to develop the disease, or who's responding to a medication at what dose, these kind of questions. So I want to thank you for giving me this opportunity to tell you a little bit about my technical field and the kind of opportunities for big data science it produces. Thank you.
Thank you very much for this very interesting presentation. I think we have time for one question. There's anyone? Okay, go ahead.
All right, David, please, thank you very much. Amazing technical information. I have a question regarding the PET scan that you indicated regarding the amyloid for dementia. So it's interesting that you can diagnose people with those potential of this disease. The question, if you discover they have this potential disease, there is any medication to prevent this disease from happening?
Yeah. The basic answer is not yet. There are some medications that are currently either in various stages of development that may slow down the progression of the disease, but nothing that prevents it at this point. But it's a very active area of research. Lots of the drug companies are just completely focused on dementia drug development. Thank you.
Thank you very much. But one brief question, like how many of the people you see would go for this test?
So it's, you know, I do it, so you can do PET imaging for clinical purposes or for research purposes. For clinical, when it's indicated, you know, people like who have dementia where they're trying, or cognitive decline, I want to know whether, so that's an easy sell. You know, it's easy for them to volunteer to do a PET study because it's going to give clinically useful information. But for research purposes, it's more complicated. So we have like a team of experts really that can speak to patients and tell them about the risks of the procedures themselves, and the relative benefits, which are not directly to them, it's more benefits to science. We have a lot of expertise in doing that and have been very successful. But yeah, it's you know, a big part of what we do is like be able to recruit research subjects that are appropriate and characterize them.
Thank you.